I'm Jing Wang, a principal investigator working in Sichuan University in China. It is a great honor for me to present our recent work about genomic insights into present local adaptation and future climate change vulnerability of a keystone species in East Asia. Global climate change is one of the largest threats to biodiversity of our times. It can cause many consequences, including loss of glacier ice, wildfires, floods, drought, and heat waves, and so on. Many plants have been standing where they are today for hundreds or even thousands of years. So we have two questions. First is how have they been able to cope with the changing conditions, like drought, flood, and heat stress over such a long time? The second question is what will happen next? Will they migrate into more suitable places or go extinction or have the ability to adapt and cope with the rapid future climate change? Recently, the advancement of sequencing technologies enable us to generate large amounts of genomic data, and the genomic data have been certified to be a good new tool for predicting future climate adaptation and maladaptation. Forest trees are keystone species in local ecosystems, and it is important to understand their response to future climate change. Temperate forests in East Asia have high biodiversity. However, compared to other regions, temperate forests in this area still lack a comprehensive evaluation of potential climate change impacts. So, in this study, we selected one of the dominant tree species in East Asia, Populus coreana, as our study system. We have two main aims. First is to investigate the genomic basis of current local climate adaptation, and second is to predict its evolutionary response to future climate change. Here is the overall workflow of this study. First, based on long-read, short-read, and high-C technology sequencing data, we assembled a chromosome-level genome assembly with very high quality. For example, the Contig N50 is 6.4 megabases, and the complete BASCO is also very high. We found that Populus coreana share the same ancient whole genome duplication with other Populus and Salix species. From the phylogenetic relationship analysis, we can see that Populus coreana is most closely related to Populus kappa, and they diverged around 2.7 million years ago. Based on the population structure analysis, we found that the 24 populations we sampled can generally divide into northern and southern groups. There is significantly isolation by distance pattern. From the demographic history analysis, we found that different populations displayed highly similar demographic trajectories. The inferred effective population sizes only differed between northern and southern groups following the last glacial maximum, around 10 to 20,000 years ago. Although we detect population structure among these populations, we find their genetic divergence is relatively quite low. The average FST values between northern and southern populations is only 0.02. After accounting for the population structure among these populations, we used two methods to detect the variants potentially involved in local adaptation. In total, we identified more than 3,000 variants to be involved in local climate adaptation, most of which are SNPs, but we also found some candidate adaptive indels and structural variants. As we can see, they are distributed across the whole genome, and most of the adaptive variants located in non-coding regions. In particular, we found a significant enrichment of climate adaptive variants in 5' untranslated regions. Moreover, we found more adaptive variants are associated with precipitation compared to temperature-related variables. When we focus on genotype environmental associations, results of specific climate variables, for example here, one temperature-related variable maximum of maximum temperature or warmest month, and another precipitation-related variable, precipitation or wettest month. We observed a polygenic pattern of climate adaptation, where we identify many well-studied genes potentially involved in adaptation to various environmental variables. If we choose adaptive variants from different genes of these two environmental variables, we can see that the adaptive variants showed very similar patterns of allele frequency distributions across different loci. We thus choose two candidate genes for further exploration. First, we selected an example candidate loci associated with variation in the maximum temperature of warmest month, which homologous to Arabidopsis HSP60, which encodes a heat shock protein that can be induced by heat stress and protect cells from heat damage. We choose one candidate adaptive SNP located in an intronic region to display the results. We can say the populations located in areas with relatively higher temperature of warmest months of the year were more likely to carry the G allele, while the A allele was more likely to be observed in regions with low temperature. There is relatively high LD found within the region surrounding the gene. The haplotype carrying the warm adaptive G allele have elevated extended haplotype homozygosity relative to the other A allele, suggesting the G allele might have experienced a weak positive selection. Moreover, to further verify the potential function of this gene, we performed qPCR to profile its expression under heat stress. We found that the genotype with the candidate warm adaptive G allele showed much higher expression than the A allele after 2 and 3 hours of heat stress treatment. All these results suggest that HSP60 is likely a candidate gene for heat stress tolerance in Populus coreana. Next, we also selected an example candidate loci associated with variation in the precipitation of wettest months. We found two tandem duplicates homologous to Arabidopsis CLL1 gene, which is a transcriptional factor involved in root system architecture. We also selected a candidate adaptive loci located in 5' untranslated region as an example to show the results. We can say that the T allele is mainly distributed in the southeast regions of the distribution that are characterized by heavy precipitation in the wettest months, whereas the C allele was almost fixed in areas experiencing low rainfall. We also observed relatively high LD in this region, and the haplotype with C allele display a bit higher extended haplotype homozygosity than the T allele. In addition, to further verify the potential function of this gene, we also performed the qPCR to profile its expression under submergence stress. Very interesting, we observed in response to submergence, individuals with TC genotype displayed enhanced expression compared to individuals with the CC genotype. Again, all these results suggest that the CLR1 is likely to be involved in precipitation-associated adaptation. Moreover, we also identify many other candidate genes involved in other climate variables, including both temperature and also precipitation-related variables. For example, here, we show that the gene homologous to PIL1 is highly associated with precipitation seasonality. Based on the established genotype-environmental relationship and the identified climate-associated genetic loci, we next want to make predictions of how populations of Populus coreana will respond to future climate change. There are two questions here. The first is how the genomic information can be used to predict future vulnerability of species under climate change. The second is how the genomic adaptation and maladaptation to future climate will differ across populations. We use two different approaches to make prediction. The first is called the risk of non-adaptiveness. Using the climate-associated variance, it first establishes a linear relationship between allele frequencies and present climates. Based on this, we can measure the shift of allele frequency required to cope with future climate conditions, which represent the risk of non-adaptiveness. At the scale of climate variables, we generally find the pattern that the risk of non-adaptiveness for temperature variables was substantially higher than those projected from precipitation-induced responses. It indicates that the higher temperature in the future is still the major factor influencing the future adaptation of Populus coreana. At the scale of populations, we find that populations located in areas with more drastic environmental changes are anticipated to have greater risk of non-adaptiveness values. Although populations in both northern and southern distributions have almost equally high values of risk of non-adaptiveness in the face of temperature changes. Southern populations displayed much higher genomic vulnerability compared to northern populations because of extreme precipitation events in the future. Another approach we use is the gradient forestry analysis, which is a machine learning method estimates the multi-locus genetic offset between current genomic composition of populations and future climate conditions along the gradient. It first determines the relative importance of various environmental variables based on the candidate climate-associated variance. We choose the 6 most important and uncorrelated environmental variables to estimate the genetic offset across the geographic distribution of Populus coreana. Similar to the risk of non-adaptiveness estimates, the genomic vulnerability estimated by the gradient forestry analysis was also found to be highest in southeastern populations near Korea Peninsula. In addition, recent studies show that the estimate of genetic offset or vulnerability can also be influenced by neutral processes such as effective population sizes. To check whether our results are influenced by these neutral processes, we examined the relationship between genetic offset and both nucleotide diversity and genetic load. We did not find any significant relationship among these factors. It indicates that our results should be quite robust to other neutral factors. So I will make a short conclusion here. We first assembled a high-quality reference genome for Populus coreana, then we make a comprehensive characterization of the genomic basis of climate adaptation. Based on these results, we can predict the spatial-temporal responses of populations to future climate change. There is already a preprint of this study online. Please read it if you want to know some more details. Last, I want to thank all the lab members and collaborators who are involved in this study, especially Yupeng and Zhiqing, who lead most of this work. I also want to thank the fundings that support this work. Thank you all.